Alrighty, so the study that we're going to look at is the Rosenzweig study, Rosenzweig et al., which was done in 1972, which is going to help us answer the learning outcome about how our environment can affect our physiology. Okay? And so basically the environment we're looking at is the environment in which the rats live, and the physiology is their brain size and how complicated their brain is. So basically the theoretical proposition or the hypothesis was that animals raised, actually I'll highlight that, animals raised in highly stimulating environments will demonstrate differences in brain growth and chemistry when compared with animals reared in plain or dull circumstances. So basically what we had was 12 sets of 3 male rats from the same litter. So what they had was that there was one litter, and they'd take 3 male rats from that same litter. They'd put one of them in a control setting. They'd put one of them in an impoverished or poor or boring or dull setting. And then they'd put one in an enriched, really fun, exciting setting with lots of stimulation. And so obviously you see in the control group, they were put in with several other rats in a normal-sized cage, several other rats of their own litter, and given sufficient food and water. In the impoverished setting, the poor setting, the boring setting, it was one poor little rat all by itself in a small cage with also sufficient food and water. And then the third sibling from those 3 male rats was put into an enriched cage, which was like a rat Disneyland, hence the Mickey Mouse, with all kinds of fun games to do, the running wheel, and all kinds of things, and a few other friends to hang out with, also given sufficient food and water. They did this with 12 different sets of the male rats. Next. Okay, so they had these 3 conditions, and they did this test 16 different times, each time for 4 to 10 weeks, without the researchers knowing which mice they were examining in the end, after the mice had been humanely sacrificed, so that there were no demand characteristics or researcher bias or expectations about which mice's brains they were looking at. The results were pretty significant, okay? How did the mice's environment affect their brains? Well, their cerebral cortexes of the enriched rats were heavier and thicker, okay? The cerebral cortex is the outer part of the brain, which responds to experience, is involved with movement, memory, learning, and sensory input. So all, smell and feeling and tasting, that all goes through the cerebral cortex. Also, increased activity of the enzyme acetylcholinesterase. Obviously, you see it looks similar to acetylcholine, which is the memory neurotransmitter. So increased activity of this enzyme. Larger neurons. So there weren't more neurons, which are the brain cells, but there were larger neurons, and a greater ratio for RNA to DNA, which is important for cell growth. These are more lovely neurons over here. And lastly, the synapses of the enriched rats were 50% larger. So bigger synapse, more space for more activity, more neurotransmission to be taking place. Okay, so remember that the learning, the command term for this learning outcome for this question is discuss. So we obviously want to consider a bunch of different arguments and different opinions and different factors when we're answering the question, not just summarizing. So a few questions to bring up and consider. Was the rat's handling different when the toys were changed in the enriched environment cage? So were they handled more? Maybe that could have enriched their brains, and it could be a confounding variable. Well, later this became somewhat unimportant because they retested it with the same amount of handling for the impoverished and the enriched rats, and there was no difference. Were the impoverished rats stressed because they were all alone, and could that have affected their brain structure? Again, they tested other stressed rats who had been under really stressful conditions like electric shocks and stuff, and their brains never showed any significant differences from any of the other brains that they looked at in this experiment. Do wild rats have even more developed brains than the enriched lab rats? And the answer is yes. So quite an interesting thing to consider, that the real-life rats, the ecologically valid rats, had even more developed, thicker, more complex brains than the enriched lab rats. So there's no substitute for nature, okay? And can we generalize any of this to humans? We'll talk a little bit about that on the next slide. And lastly, Olson et al. Did some research on, what about the enriched environment made the mice's brains bigger and more complex. And they kept trying to isolate different things within those enriched cages. So, was it the company of the siblings, or was it this specific toy? And what they found was that the exercise wheel was the most significant factor in the whole cage. And so they're wondering, maybe it's not just, stimulation and enrichment, but specifically voluntary exercise. Was it just the running wheel that made the rats' brains grow? And, they found that in some situations, yeah, it really was just the running wheel. So Olson can talk about the role of voluntary exercise compared to just enriched environments. Okay, our last slide just talks quickly about the real-life application and the human evidence to support this. So why does it matter? Who cares? What's the big picture? What are the implications of this research? And that would be improving memory and researching on how we can improve our memories and the functioning of our brains, and also preventing memory loss in old age, and also researching the effects of malnutrition on intelligence. So malnutrition being an impoverished environment, or an effect of an impoverished environment on intelligence in humans. And lastly, we've got, what is some human evidence that we can use to back this up to show that, yeah, maybe it can't be generalized completely, rat to human, but, there could be some similarities. And the evidence to back it up, to support this, would be that brains of humans who have died naturally, that have been examined, do show that the more skills and abilities a person had when they were alive, the more complex and heavy their cortex was. So those skills and abilities being anything from playing a sport or an instrument to maybe being especially gifted in computer programming or something like that. And lastly, examinations of blind people's brains after they die show thinner, simpler, and less developed cortexes in the visual regions, in the visual sections of the brain. So again, they weren't getting the visual stimulation because they were blind, and that part of their brain was less developed.